penicillium life cycle shows mainly sexual reproduction and asexual reproduction. During the asexual reproduction, the asexual reproduction occurs in the favorable conditions of the mycelium. During the asexual reproduction or during the favorable conditions of the penicillium, they produced aerial stalk-like structures called conidiopores. On the conidiopores, rami, on the terminal part of rami, medullae, on the terminal part of medullae, sterigmata. Sterigmatas are in flower vase-like structures. From these sterigmatas, conidiums are developed. The conidiums are spheroidal condition. They contain haploid nucleus. They are developed in a basipetal manner. Basipetal manner means first developed conidiums are far from the conidiospores or far from the sterigmatas. Early developed conidiums are above the sterigmatas. At matured stage of these asexual reproduction, the conidiums are leave from the sterigmatas and they are landing on the moisture surface. They are germinated into new vegetative mycelium. It is the continuation cycle during the favorable conditions. During the favorable conditions. During the unfavorable conditions. During the unfavorable conditions, the penicillium vegetative mycelium participated in the sexual reproduction. During the sexual reproduction, the vegetative mycelium of penicillin produced young ascogonium. The young ascogonium is in club shaped, it filled with more number of haploid nuclei with cytoplasm. At this stage of ascogonium, near these ascogonium from the remaining penicillin mycelium, ribbon-like structures are developed. The ribbon-like structures are called antheridial branch or antheridium. Ascogonium is a female reproductive organ of the penicillium. Antheridium is a male reproductive organ of penicillium. The antheridium filled with more number of nuclei with cytoplasm. At matured stage of ascogonium and antheridium, around the ascogonium, antheridium is arranged in a spiral manner. The antheridium spirally arranged around the ascogonium at the matured stage. Finally, at the matured stage, the antheridium terminal part contact with ascogonium cell wall. By this contact method, cell wall is ruptured between the antheridium and ascogonium. At this time, from the antheridial branch, only cytoplasm enter into ascogonium. That ascogonium cytoplasm fused with antheridium cytoplasm in the ascogonium. This process is called plasmogamy. After plasmogamy, the ascogonium convert into cellular portions. Before the plasmogamy, we observed the ascogonium in the cenocytic position. The cenocytic position with more number of haploid nuclei of ascogonium. After plasmogamy, the ascogonium convert into cellular portions. Each cell contains 2 nuclei. This stage is called binucleated stage of ascogonium. The binucleated stage of ascogonium produced lateral filamentous-like structures. From each binucleated cell, lateral filamentous-like structures are developed. They called ascogenous fungal hyphae. The ascogenous fungal hyphae is in branched structure. Each cell of ascogenous fungal hyphae containing 2 cells, 2 nuclei. It is also called binucleated stage. The ascogenous filamentous structure also in binucleated structure. From this binucleated structure, one nucleus convert into ascus mother cell. One binucleated cell convert into ascus mother cell. The ascus mother cell containing 2 haploid nuclei. The 2 haploid nuclei belongs to only ascogonium, not from antheridium. The ascogonium 2 haploid nuclei present in the ascus mother cell. They are fused to form diploid zygote. The diploid zygote is also called diploid nucleus. This stage is called karyogamy. Karyogamy done by the similar types of nuclei fusion. Same type of nuclei fusion from the ascogonium. If diploid zygote is formed, it participated in the reduction division or meiotic division. to form 4 haploid nuclei. The 4 haploid nuclei again participated in the mitotic division to form 8 haploid nuclei. See in this diagram only 7 given, but 8 haploid nuclei are present. That 8 haploid nuclei are developed into ascospores. The ascospores are in pulley wheel structure. Around these ascospores, ascus, ascogonium, ascogenous mother cells, and ascogenous filaments, and ascogonium and antheridium. New fungal hyphae is covered as a cover-like structure. Finally, it looks like a fruit-like structure. The fruit-like structure is called clistothecium. Below the clistothecium, ascospores, ascus mother cell, ascogenous filaments, ascogonium, antheridium. These all are present below the clistothecium. At matured stage, the pulley wheel ascus or ascospores comes out from the clistothecium. They are absorbed moisture from the surface. They are developed into germination. The germination of ascospores develop into new vegetative mycelium. It is the sexual reproduction in the penicillium. Now we observed the penicillium, vegetative penicillium, mycelium in haploid condition. The haploid condition penicillium produced aerial stalk-like structure during the favorable conditions called conidiopores. From these conidiopores, conidias are developed. The conidiopores are haploid in condition. Conidias are in haploid condition. They are germinated, again develop into new haploid mycelium. During the unfavorable conditions, they participated in the sexual reproduction. During the unfavorable condition, the vegetative mycelium of penicillin produce ascogonium, it is the female reproductive organ, and antheridium is the male reproductive organ. Antheridium, ascogoniums are participated in the plasmogamy. Mean antheridium cytoplasm enter into ascogonium. They called plasmogamy. After plasmogamy, male nuclei present in only antheridium, female nuclei present in the ascogonium. Ascogonium containing different types of paired nuclei. They are developed into binucleated stage. After binucleated stage, lateral side, ascogenous fungal hyphae are developed. From the ascogenous fungal hyphae, penultimate cells are developed. They are called ascus mother cells. Ascus mother cells containing haploid nuclei. 2 haploid nuclei are present. The 2 haploid nuclei are participated in the karyogamy to form diploid zygote. The diploid zygote develop into young ascus. The young ascus develop into young ascospores by the meiosis division. If meiosis division occurs in the clistothecium, ascus mother cell containing diploid zygote, it participated in the meiosis division, it developed into asci or ascospores. Ascospores are germinated and they are developed into new vegetative mycelium of haploid condition. See, in this stage, haploid stage is present, haplophase, and dikaryophase also present, diplophase also present. Haplophase, bicelled phase, diplophase. These are present in the life cycle of the penicillium. Haploid phase, diokinophase means binucleated phase. Binucleated phase after plasmogamy. And after plasmogamy, binucleated cells are developed into ascus mother cells. The ascus mother cells are participated in the karyogamy. They are developed into diplophase. Diplophase means diploid zygote is formed in the ascus mother cell. It develops into young ascus. The young ascus develop into clistothecium. In this clistothecium, ascus mother cell participated in the meiosis division. They develop into haploid ascospores. The haploid ascospores germinated into the new vegetative mycelium of penicillium. It is the life cycle of penicillium.